Video goes along with section 12.4, the end of chapter 12, and then the beginning of chapter 13, 13 plus 1.1, to basically the first page of 13.2. 13.2 has to do with DNA replication, and we're going to spend more time on that. So 13.2 will be another time. What we are going to focus on is chromosomal mutation. So chapter 12 is about chromosomal basis of inheritance. So talking about chromosomes, linked genes, things like that. That's why 12.4 will have to do with mutations that happen on a chromosomal basis that have to do with the whole chromosome. And then we'll talk about DNA structure. So chapter 13 is the molecular basis of inheritance. So how does genetics work at the molecular basis? And what I want to emphasize is all of chapter 12 to talk about chromosomes and all of chapter 13 to talk about DNA. Mendel had no idea of either of those things. He didn't know about DNA or chromosomes, and he was able to deduce all of his genetic laws and principles without knowing DNA, which is the material of inheritance, and chromosomes, which are the units of inheritance. So, one type of chromosomal mutation is alteration of normal chromosome number, and that is called and it happens through nondisjunction. So one type of chromosomal mutation that can occur is if you have too many or too few chromosomes. For example, if humans normally have 46, if they had 45 or 47, 44 or 48, too few or too many chromosomes. So, how does that happen? Well, that happens in the process of nondisjunction. Sometimes errors happen in meiosis. So meiosis is where we're making our gametes that result in gametes with too many or too few chromosomes. So sometimes gametes will have 24 or 24 or 22 chromosomes instead of the normal 23. And that happens because of nondisjunction and meiosis. And gametes that don't have enough chromosomes or have too many are called aneuploids. So too many or too few chromosomes is called aneuploidy. Sometimes these gametes can create viable offspring. So if I have a chromosome that has, let's say, or a gamete that has 24 chromosomes instead of 23, and it combines with a normal gamete that has 23 chromosomes from the other partner that is making this offspring, the offspring would then have 24 plus 23 or 47 chromosomes. And sometimes those offspring can be born and grow normally, or I just mean don't end up having their life cut short or not even being viable. And so you could end up with a human that has 44, 45, 46, 47, 48 chromosomes. Aneuploidy occurs when either homologous chromosomes don't separate in metaphase of meiosis 1, or sister chromatids don't separate during metaphase of meiosis 2. So, nondisjunction can happen in two ways. In meiosis 1, when homologous chromosomes are lining up across from each other on the metaphase plate. What can happen sometimes is both of the homologous chromosomes get drugged to one side of the cell, of the new cell, and that means the other side only gets one fewer. They don't have one over here. So in meiosis 2, you get a gamete with an extra chromosome and gametes that have one too few chromosomes. So this is how one way nondisjunction can happen. Another things can go normally in meiosis 1. However, in meiosis 2, sister chromatids don't get pulled apart in sometimes. And so you're resulting with some gametes that have one too many chromosomes. Some gametes that have one too few chromosomes. And you also get some gametes that have a normal number of chromosomes. So, if nondisjunction happens in meiosis 1, in metaphase 1, you can get gametes with too many or too few. That's it. If nondisjunction happens in meiosis 1, you cannot get a normal number of chromosomes in a cell. If nondisjunction happens in metaphase 2, in meiosis 2, you can get all three, too many, too few, or normal. Fertilization between an aneuploidy gamete and a normal gamete results in either trisomy or monosomy of certain chromosomes. So, for example, if a normal gamete fertilizes this gamete that has 2 copies of one chromosome, that offspring will have 2 copies of that chromosome from this parent and 1 from the gamete that is normal. So they'll have 3 of this chromosome instead of 2. That's what's called trisomy. However, if they get a normal gamete gets fertilized by one that has 1 too few chromosome, then the resulting individual will be missing a chromosome, only have 1 copy of that chromosome, will have monosomy. The second type of chromosomal, going with the whole chromosome, chromosomal mutations is the alteration of chromosome structure. So, how that chromosome is laid out. The structure of chromosomes can be altered by mutations or mistakes during meiosis. These structural changes come in 4 types: deletion, duplication, inversion, and translocation. And this is a good picture to show you the differences. In deletion, our DNA on our chromosome gets broken, and a gene or a stretch of DNA is taken out, and the chromosome reforms or religates together, and we're missing some DNA. Part of it gets deleted. Kind of the opposite, sometimes breaks in our DNA can happen, and this segment gets recopied and added to the chromosome before it gets ligated or stitched back together. And so we can have twice as much of certain genes. An inversion can happen, where literally our chromosome breaks in 2 spots and the DNA that is in that stretch is flipped in the opposite order and ligated back together, so it's inverted. And last but not least, 2 non-homologous chromosomes during crossing over in meiosis 1 and metaphase 1 can switch unequal segments from non-homologous chromosomes. So here we have gene AB, and it can switch in crossover with segment MN and O from this chromosome. And so we have a recombination, a translocation of genetic information. Deletion and duplication are especially likely to happen in crossing over when non-sister chromatids exchange unequal lengths of DNA. Human genetic disorders that are associated with chromosomal alterations? Well, 2 examples of non-disjunctive mutations that occur in human genetic disorders. One is trisomy 21, or Down syndrome, and really hard to see because I unfortunately blew this up too big, but let's see. We'll start on this slide. But down here is a karyotype of someone with Down syndrome, and they have 2 copies of every chromosome, except down here they have 3 copies of the 21st chromosome. That is what causes Down syndrome. They have 3 copies of the 21st chromosome because A non-disjunctive event happened, either meiosis 2 or 1, and there was a gamete that eventually fertilized the egg cell or in the egg cell itself, and it had one too many chromosomes. Now, one too many was the 21st chromosome. In addition, there's an aneuploidy of sex chromosomes. So some disorders, like Klinefelter syndrome, you can have too many or too few sex chromosomes. So in Klinefelter's, this individual has 2 X chromosomes and a Y. How did that happen? Well, a non-disjunctive event happened here, but it was in the X chromosome. So the extra chromosome was an X chromosome, a sex chromosome, and it caused Klinefelter syndrome. 2 examples of chromosomal alterations that cause genetic disorders is Cri-du-Chat's. It's a genetic disorder caused by a deletion on chromosome 15. So a deletion happens. Part of it gets cut out. And chronic myelogenous leukemia happens as a result of translocation of a large part of chromosome 22 with the tip of chromosome 9. So these 2 parts are going to switch places, and this occurs. And that results in some abnormal blood proteins being created, and a type of leukemia results. Like I said, very hard to treat these things. They're usually chronic, can't do anything about them because they're genetic. It's in your genes. We can't go and flip these things back around, and we can't take out a chromosome in all your cells. It doesn't work like that. Alright, so that's kind of the end of chapter 12. So now we're going on to chapter 13. This is talking about the structure of DNA, inheritance, the molecular inheritance of DNA. So molecular inheritance through DNA. So the first question is, what is the material of genetic inheritance? Big question that was left by Mendel's work. What is passed on? What exactly is passed on from parents to offspring that carry the genetic information, these traits? Mendel didn't know about DNA or chromosomes, so he couldn't answer that. Scientists after Mendel struggled to determine what chemical or what exactly was the material that's passed on from parents to offspring. Frederick Griffith discovered that there was a chemical that passed on genetic information. He did that by investigating bacteria and bacteriophages. A bacteriophage is just a virus that infects bacteria. And so he cultivated a living bacteriophage virus that was virulent or deadly, and he injected it into a mouse. And like you would suspect, it killed that mouse. He also raised a second strain of bacteriophage virus that wasn't deadly, and injected a mouse with those, and the mouse was just fine. Third, he killed with heat the virulent deadly strain, and when he injected it in the mouse was fine. This was dead. But what he did then is he combined some heat-killed virulent deadly strains of bacteriophage, a virus, with a living, not deadly strain. And when he injected a mouse with that cocktail, it died. And what did he conclude? That genetic information for making the toxin in the dead virulent strain was passed to the avirulent strain through some chemical substance. And he called this process transformation. And so, by doing that, he proved or supported the fact that there was a chemical substance that carried the genes for producing this poison that was transferred from this bacteria to this bacteria. Later on, two experimenters named Hershey and Chase proved that it was DNA that was the substance of genetic inheritance. And basically what they did is they took bacteriophages, viruses, then viruses are made up of protein and DNA. And what they did is in one group of viruses, they incubated them with radioactive sulfur, which became incorporated in the protein of the DNA. And so when the virus injected its DNA into the bacteria, they blended it together to kind of separate the virus and the bacteria, and then they centrifuged it. The bacteria was heavier, the virus was lighter, and so the virus was on the top. And they measured the radioactivity. It was all here at the top. All here at the top where the protein in the virus was, and none of it was down here in the bacteria or the injected DNA in the bacteria. So they determined that since this bacteria would be receiving whatever chemical the virus was injecting, that was going to be what was the material of inheritance. They did a second batch with radioactive phosphorus. Now, phosphorus was incorporated in the phosphorus backbone of the DNA, so when they followed the same procedure, the radioactive DNA became incorporated into the bacteria cell. So they saw the bacteria cell was radioactive and the virus solution, the protein solution, was not. And that's how they determined, yep, it's the DNA that is material of inheritance. So once we determined what the material of inheritance was, we need to figure out what the structure of DNA is. Well, scientists already knew that DNA was a polymer of nucleotides. It was made up of phosphorus and sugar backbone ribose and nucleotide and nitrogenous bases, but they didn't know the overall 3D structure. Chargaff was really important because he determined that the amount of adenine and thymine in a single sample of DNA was always equal, and that the amount of cytosine and guanine were always equal. So the number of adenine bases in a long piece of DNA was always equal to the number of thymine bases in that same piece of DNA, and that also the number of cytosine bases in that long strand of DNA was also the same as the number of guanine. This was what was already known. Two scientists named James Watson and Francis Crick tried to build molecular models like this one to figure out DNA's structure. So they tried to take this information and make a 3D structure hypothesis, and whichever one worked out the best and was the most consistent with experimental evidence, that must be what its structure was. And they made a first attempt that was wrong, looked like this. It was incorrect. So they went back to the drawing board, and they, sneak around another university, King's College, and they saw this picture that was made by Rosalind Franklin, and they basically stole her work, her information. They used it to update their model and didn't give her any credit. And this picture is an X-ray diffraction image. So it's basically a picture of DNA made by X-rays bouncing off the non-absorbent parts. And they could see in this picture this clear X shape. And that clear X shape told them that DNA was formed in a double helix. And with that, they were able to make their new model like this. And they were awarded the Nobel Prize for determining the structure of DNA by stealing Rosalind Franklin's work, pretty much. She never received the Nobel Prize because it can't be awarded posthumously. However, her colleague Wilkes did. So James Watson, Francis Crick, John Wilkes all received the Nobel Prize, and she was left out. But from it, we were left with this double helix structure of DNA. So this is what DNA, the structure, looks like. It is a winding staircase, what we call a double helix. There are 11 rungs or base pairs. These are what we call a base pair. There are 11 of them for every 360-degree turn. Every 11 of them for every 360-degree turn. DNA is made up of monomers. It's a polymer made up of a monomer. And the monomer is a nucleotide. And one nucleotide is made up of a deoxyribose sugar, a phosphate group that's attached to the sugar, and a nitrogenous base that's attached to the sugar, like this. So it's made up of repeating units of these nucleotides over and over and over again. And one nucleotide on one strand, it's double-stranded. They have two strands. One nucleotide is joined via hydrogen bonds to another nucleotide on the opposite strand via hydrogen bonds between their bases. And the bases that hydrogen bond together are called base pairs, or pair up. A, or adenine, always pairs with T, thymine, and C, cytosine, always pairs up with G, guanine. That's why Chargaff noted A and T numbers in DNA were always equal, because they were the only ones that could pair up, and C and G were always equal. They were the ones that could pair up. So you'll notice that some of the bases, like A and T, A and G, are made up of two rings, and we call those bases purines. And C and T are made up of one ring, and they're called pyrimidines. We also notice that DNA is anti-parallel. What that means is that it has one direction on one strand and the other strand is going in the opposite direction. And we call that 3 and 5 ends. And we do that by counting the carbons on the sugar molecule. Starting to the right of the oxygen, so 1, 2, 3. This is the third, so we call it the 3 end. 4, 5. This is the 5 end. So this DNA, all of the 5 ends are pointing this way. All of the 3 ends are pointing down. It's just the opposite on the other strand. 1, 2, all the 3 ends are pointing this way, and all the 3, 4, 5 ends are pointing this way. That's what a 3 and 5 end means, and that's what it's meant by anti-parallel. The strands, the 5 end on one strand is the same direction as the 3 end on the other. So this is what Watson and Crick were able to deduce. They were also able, here is a picture of the bonding between adenine, thymine, cytosine, guanine. You'll notice cytosine and guanine can have 3 hydrogen bonds between it, and A and T can only have 2. So just a picture. What they also deduced, Watson and Crick, was that DNA, its structure lended itself to replication. So the unique base pairing pattern in DNA, in its double-stranded anti-parallel nature, immediately suggested a mechanism for self-replication. DNA has to replicate, remember, during mitosis and meiosis in the S phase? And it does so by basically unzipping itself, coming apart, and synthesizing new complementary DNA to both of the strands. So we end up with two strands of DNA, half of it being new. Since we only have to synthesize half new DNA from a strand, we already have a template strand. It's called semi-conservative because we conserve half of the DNA. Meselson and Stahl confirmed that DNA replication was semi-conservative by doing this experiment. Alright? In one beaker, they cultured bacteria in a medium with radioactive nitrogen, and that DNA sample was centrifuged after the first replication of that DNA. And and bacteria. And the second one was transferred or grown with radioactive nitrogen-14, which was lighter. And it was put aside on its second replication. And what they noticed is there was these separations. There was a band where there was less dense, come from the nitrogen-14, and there was also a band from the more dense, 15. The only way that could have happened is if part of this 15 was incorporated with part of this 14 on the template strand, so being semi-conservative. And that's how provided scientific evidence that DNA replication was semi-conservative. So next time we're going to get into the mechanisms, what's happening in DNA replication. But this is it for today.